Welcome to All the Best, an interview series with UNL ORCA awardees, where UNL means University of Nebraska-Lincoln, and ORCA is the Outstanding Research and Creativities Award, which is UNL's top award. In Gerda Lerner's book, Why History Matters, she explains the life stories give meaning to the present and the future because we identify what is important to us. And she says, our stories show how we are interconnected in a particular time and place. As for this interview series, ORCA awardees have experiences that reach back more than a half a century, which will give us a personal view of how the best research emerged from UNL. It is my pleasure to introduce today's interviewee, Dr. Donald Paul Weeks. He was awarded the ORCA in 2014, a year before he retired. After I provide a quick summary of his life and career, Don will tell us about his time at UNL, including his award-winning research. Don grew up on a small farm south of Terre Haute, Indiana. In his youth, he participated in 4-H programs that must have sparked an interest in science because he earned his bachelor's degree from Purdue University in 1963. Purdue is about 90 miles north of Terre Haute. Don's doctoral work was at the University of Illinois in Champaign-Urbana for a project about protein synthesis during maize germination. Next, he did postdoctoral work at the Fox Chase Cancer Center in Philadelphia, where his research concerned the mechanisms by which protein synthesis was regulated in eukaryotic cells, and I should say is regulated. After the postdoc ended, he remained at Fox Chase as a researcher from 1970 to 1981. From there, he moved across the country to Sandoz Agro Research Institute in the Stanford University Industrial Park in Palo Alto. He was a principal scientist at Sandoz for 7 years, where he worked on plant molecular biology. In 1989, he joined the University of Nebraska-Lincoln as the first director of the Center for Biotechnology. His research and tenure home was in the biochemistry department. With that, I want to welcome Don to the interview.
Thank you.
What was your impression about the UNL campus when you saw it for the first time? And what were your thoughts about the campus when you retired?
Okay. So when I first came to the campus, I was impressed that it was what I saw as a typical Midwestern land-grant university. As you mentioned, I went to Purdue University and the University of Illinois. And so the Lincoln campus was not a lot different than those, although I was struck with the lack of uniformity in architecture. I was also a little bit disappointed in the landscaping. And that for an agricultural university, I thought it wasn't quite up to snuff. So that all changed, though, very soon after I arrived, primarily through the efforts of Kim Todd and her associates and sprucing up the campus beautifully. So I think a lot of progress was made in that regard, and it's now a very pleasant university campus to visit and to work on.
And yeah, so where were you first located when you started at UNL?
That's an interesting, a complicated story. So my academic home was the biochemistry department, which was in Biochemistry Hall on East Campus, which is where Massengill Hall is right now. But I was also the director of the Center for Biotechnology, and our offices, actually the first one was in the Animal Science Department. They gave us office space, and then about a year later, we moved into new renovated space in Mantor Hall on City Campus. A nice renovation. We were there for a number of years until the Beetle Center opened, and we moved the Center for Biotechnology, my offices, and all of the core research facilities into that building.
Great. And so when you came to UNL, describe for us your research area and why that was important.
So I had a number of research areas, actually. Uh, my fundamental research was in molecular biology, and in particular, I was interested in using algae, Chlamydomonas reinhardti, as a model system to study gene regulation in eukaryotic cells. But on the practical side, I also wanted to set up a plant genetic engineering project, which ultimately I did, which was the project that led to my ORCA award in developing dicamba-resistant crop plants. So I also had a side project with a model system, Arabidopsis thaliana, a small little plant that grows rapidly, goes seed to seed in a very short period of time. So several different areas of interest.
Great. So then tell me how you recruited graduate students and postdocs in those earliest years.
Yeah, so I had several postdocs and graduate students. I had, when I was at the Fox Chase Cancer Center in Philadelphia, I had a number of postdocs. And also, when I worked in industry, I had an NSF, National Science Foundation grant during that period and had postdocs working with me. Coming to the University of Nebraska-Lincoln was my first experience with a large number of graduate students. I'd had one before at Fox Chase because I had an appointment to the faculty at the University of Pennsylvania at that time as well. But UNL was my first experience in training a number of graduate students. And that was really a great experience. Challenging at times, but rewarding in seeing the growth of those students. And then I've been blessed with a number of great postdocs over the years who've gone on to really great careers.
Yeah, great. It occurs to me that you came from a non-academic background, joined UNL. This was your first research university?
Yes, so the Fox Chase Cancer Center was a very well-known fundamental research laboratory. Could have been called the Institute for Cell Biology, in addition to the Institute for Cancer Research. Well, there were about 43 members. 8 of those members were members of the National Academy of Science. 3 went on to win Nobel Prizes.
Wow.
So I was there for 15 years, terrific place to work. But I saw an opportunity to use my knowledge and background in molecular biology to to improve agriculture. And at that time, the biotechnology industry was just beginning to take form. And there were a number of companies, especially on the East Coast or West Coast, that were specializing in plant molecular biology. And that was my area. So I found one in Palo Alto that was called Zoecon, a small startup company that I really liked. They already had some products on the market in insect control and weed control, but wanted to start a plant molecular biology group. And so I helped with that effort. And we did a fair bit of basic research there, as well as some applied research that led to some patent applications and some products ultimately getting into the field.
And what type of cells did you work on at Fox Chase?
So in Fox Chase, I first worked as a postdoc with Abraham Marcus, who was had just established what was called the wheat germ or wheat embryo in vitro translation system. So I started out studying the mechanisms by which cells can make proteins, how messenger RNA attaches to ribosomes, all of the initiation factors that are required for that. And in particular, my first project was studying what's called stored messenger RNA. Turns out that seeds, as with some mammalian systems, when they go quiescent, they store messenger RNA so that when times get better and it's time for action, those messenger RNAs are readily available to the ribosome. So I studied where those messenger RNAs were, how they attached to the ribosomes to begin seed germination. And that's a bit of a carryover from the work I did as a graduate student at the University of Illinois.
So after coming to UNL, did the research run smoothly, or did you come up against some unexpected result that changed your direction? How'd that go?
When did research ever run smoothly? There certainly were challenges. For one thing, I came from industry, and although I had NSF grants, the only grant I had was to support my fundamental research with algae. So fortunately, I ran into Bob Spreitzer, who also used Chlamydomonas as a model system, and we talked about potential projects. And one that struck my fancy was a mechanism in algae to concentrate carbon dioxide to enhance photosynthesis. And so that's the project, the fundamental project that I homed in on. But I spent much of my career developing new methods to study biology. And so one of the things early on that I did was to develop a library of genes from Chlamydomonas that could be used to isolate individual genes. And that turned out to be very valuable to people throughout the world. And so, and that also facilitated a number of projects that we had. Then, as I mentioned earlier, one of the things that I started was a project to try to find a mechanism by which we could make crop plants resistant to the herbicide dicamba. And that's, again, the research that led to my ORCA award. And I'll explain that just very quickly here. What had happened in 1996 was that Monsanto released what are called Roundup resistant crops, corn and soybeans, that allowed the farmers to spray their crops with Roundup. And Roundup kills everything, every plant that it's applied to, well, almost every plant. And the farmers loved this. It was a terrific godsend for them in weed control. And so very quickly, most of the farmers in North America and other parts of the world adopted this technology and used it widely, indiscriminately in some regards, because after continued use, year after year, there were some weeds that developed resistance to Roundup. And interestingly, most all of those were broadleaf weeds. And I was familiar with the fact that the herbicide dicamba kills those kinds of weeds. And so I was interested to know if we could develop crop plants that were resistant to dicamba, would grow normally, produce normally, and yet the farmers would be able to kill these very difficult to control weeds. And so we inaugurated that research. And the way we did this was we first searched the literature and found that there were bacteria that would grow on dicamba as a sole carbon and energy source. So we obtained those from Dr. Doug Cork at the Illinois Institute of Technology, and found that we could break those bacteria cells open and that they would degrade dicamba in the test tube. So that allowed us then to use biochemistry and molecular biology techniques to ultimately isolate the genes that were responsible. And I say genes because it turns out that the enzyme that breaks down dicamba in the bacterium that we used is a three-component enzyme that has three separate genes that codes for those three components. That was a devilishly difficult set of experiments to do because of the three-component nature. But ultimately, we were able to isolate and all three enzymes, sequence the amino acid sequence of those enzymes, then use that information to clone the genes. And it's that technology that we patented. Along with that, we did genetic engineering of those genes so that those bacterial genes could be expressed in crop plants. We had a number of collaborators with that. Pat Herman in my lab was a real help in moving that project forward. We had close collaboration with Tom Clemente in the Plant Transformation Laboratory and a number of people on campus, especially in regard to doing some of the basic biochemistry analysis. We ultimately, with the help of Mark Wilson, for instance, went on to crystallize the dicamba monooxygenase gene or protein that's one of the three components. One of the key things that we did was notice that one of the components, for ferredoxin reductase, looked very much, this bacterial reductase looked very much like the reductase in the chloroplast of higher plants. So we decided to give a try of putting the dicamba monooxygenase gene in the nucleus of the plant cell, but attach to that gene what's called a transit peptide sequence that would transport the dicamba monooxygenase into the chloroplast. And it turns out that worked. So that meant instead of putting three genes into the plant, we had to do only one, which makes life a lot easier for us and the plant breeders that work with those genes, or that gene in this particular case. So that set of projects took several years, but we were able to patent that technology and have the opportunity to defend those patents in the court of law, which was an education in and of itself.
I want to go into I'm a biochemist, as you know, and how did the bacteria acquire these genes? Just for people who don't know anything about antibiotic resistance or insecticide or herbicide resistance, if you know the evolution of those three genes, where do you think the bacteria got them? Sounds like the one came from plants.
Yeah, so it's an interesting thing. I'm not a microbiologist, well, kind of, sort of, but not a classical microbiologist. But I've learned enough to be really amazed at the versatility of the microbial world. They can take advantage of almost any carbon energy source that's out there. So, and they've developed a repertoire of genes and enzymes that allow them to explore new possibilities. So we actually, the bacterium that we used was isolated not at random throughout the world, but Doug Cork, who found this bacterium, went to the manufacturing site for dicamba in Texas. And part of any manufacturing plant like that is a facility to catch rainwater. Water. So if anything drips off of the facility, it's caught in this pond and held for a while. Well, not surprisingly, there was some dicamba in that pond. And in retrospect, not surprisingly, there were bacteria that developed ability to use that dicamba as an energy and carbon source.
Mm-hmm.
So what's amazing to me is just how efficient the enzyme that we ultimately isolated was. When we genetically engineered the dicamba monooxygenase gene into crop plants and other model plants as well, it gave amazing levels of resistance. Normally, if you can get a tolerance to 2 or 3 times the normal level of herbicide that a farmer would use, that's adequate. We had well over 100 times tolerance to this herbicide from this one gene. In fact, the herbicide dicamba doesn't come as a pure chemical. It's formulated with other chemicals to allow penetration into the plant and so forth.
Mm-hmm.
So we could not give enough herbicide to the genetically engineered plants to kill them.
Wow.
What we were doing was killing them, but the killing was due to the solvent that was carrying the dicamba into the plant. Because if we took that same solvent and took away the dicamba and sprayed it on plants, we saw the same symptoms. And in fact, after giving some of these plants these very high doses and the plants looking almost dead, if we left them in the pot, a new shoot would come up and those plants would grow normally. So just an amazing level of resistance was conferred, which was unexpected.
Yeah. Well, that's totally amazing. So let's see. While you were doing this, you mentioned that you created some molecular biology tools. So tell me about your most cited papers and the key points.
Yes, so it turns out that the most cited papers are from our most recent research before I retired, and that was with gene editing. Gene editing is a technique by which you can use various techniques to go in and modify the sequence of a particular gene, one out of one set of DNA sequences out of billions. The system that we initially tried was called a zinc finger nuclease system, which was really tough and really non-specific. Then we went to what's called a Talon system, which worked fairly well, but was very complicated to work. And then the CRISPR system for gene editing was developed in 2012. And the minute that I saw that paper in Science, we went over to that system and to adapt it for use in higher plants and algae. And it worked beautifully. It took a while to get it to work well, but we were able to use that system to modify model plants like Arabidopsis, tobacco, but also crop plants like soybeans, sorghum, corn. And our most cited paper is a paper that showed the basic techniques used that could be used to use that technique, CRISPR, in model plants and in crop plants. And then we had follow-up publications on the practical use of that technology. So one that got a lot of attention was one in which we took an oilseed crop and changed the oil composition of the plant to produce an oil that was much like olive oil. So, and that changed the composition from 15% oleic acid to over 50% oleic acid. So I regret that those techniques came toward the end of my career because there are so many things that can be done with that technology and are being done now with that technology, and I don't get to play in that game anymore.
But it sounds like you were the pioneer, right?
Uh, of sorts, yeah.
Yeah.
That's one of the things I really, um, some people ask me what was my career, and I say to them, I had several careers. My expertise evolved dramatically over the time that I was a scientist. And that's a good thing because if you don't, you're out of business.
Yep. So let's see. Let's then go back to you being the director of the Center for Biotech. And I think the bio that I gave showed you were really thinking about the relationship between academic research and commercialization.
Mhm.
So what is it that attracted you to become the director of the Center for Biotech at UNL?
Yeah. So just as I was attracted to industry because I saw the opportunity to use biotechnology to improve agriculture. While I was in industry, I saw that a few universities were training people in this area and taking advantage of it, but not very many, in my opinion. So I started looking for opportunities. And it turns out that one of my postdocs, David Marks, was a graduate of the University of Nebraska. And he had trained here in Lincoln, and he had friends here. And he came to me one day and said, "Hey, the University of Nebraska is thinking about setting up a biotech center and is looking for a director. Are you interested?" And I said, "Well, I'll take a look." And I knew relatively little about the University of Nebraska at that time. I knew a few people here, but not very well. So I did some exploring. And indeed, what really impressed me was the fact that Nebraska had, under Kay Orr's leadership, had set up the a Nebraska system for encouraging high technology development in the university, the Nebraska Research Initiative. And that offered a fair bit of money for the Center for Biotechnology. And so there was money to do things with. There was also plans underway for a new science building that would include the Center for Biotechnology. That building ultimately became the Beetle Center. So one of the that was very attractive. Plus the ability and the money to add new staff to the university that were experts in new high technology in the biotechnology arena. So I think in terms of having an effect on the University of Nebraska per se, that my contributions in helping to set up that program, getting it running, to offer core facilities so that scientists had the latest technology available to them at a cost they could afford, was very important in changing the University of Nebraska in a significant way. So I'm quite happy with what happened in that regard.
How many of those core facilities were in place before you started, and then how many did you help
Yeah,
launch?
so there were three in place. One for DNA sequencing, one for antibody monoclonal antibody formation, and then a protein purification facility of sorts. So we developed a number of new ones. Interestingly, most of those facilities have phased out because the same services became commercially available at lower cost than we could run the facility. So most of those set down. But having those core facilities was key to attracting new faculty who needed those to run when those services were not available commercially at the time. Yeah.
Yep. No, that was very exciting. All right. So my next question is, did you publish papers that you thought were super good, but are not among your most highly cited, or ones you think are important that are not highly cited? The relationship between importance and citations.
Yeah. So one always is surprised that some of one's papers that you think are super important don't get much notice, and vice versa, that some that you think are okay get a lot of attention. And that's typical in the world of science. As we mentioned earlier, my most highly cited papers have to do with the use of CRISPR in plants. That's not terribly surprising because, as I said, I think this technology is going to have a tremendous effect on plant biology and plant biotechnology as well. So one of the papers that I think was a real breakthrough was the use of cloning techniques to find the cause of a mutation in the carbon concentrating mechanism in Chlamydomonas reinhardti. This was there was a mutation called CIA5 that had been found a number of years ago that prevented the Chlamydomonas to acclimate to very low CO2 conditions. Normally, when you put Chlamydomonas under a very low CO2 conditions, the carbon concentrating mechanism is activated, and the cells can live decently. But if that mechanism doesn't function, the cells die. So this mutation was found as one that led to cell death in Chlamydomonas, and it was seen that the enzymes and all of the systems that are turned on when the carbon concentrating mechanism is activated were not turned on. And so this became suspected to be a regulatory gene, one that regulates whether other genes get turned on or turned off. So that was key to studying the carbon concentrating mechanism. So my laboratory went after that gene. We were able to clone it, sequence it, learn lots about how it functioned. And we published this in the Proceedings of the National Academy of Science, which is a high-ranking journal. And people who studied the carbon concentrating mechanism cited it, but no one else. And to me, this was a terrifically important gene that just didn't get much attention. So a big surprise for me.
Yep. Tell me about the special activities that a researcher has, like editorships or reviewing stuff, the process of patenting, any companies you started or books that you co-edited or wrote in?
Yeah, so I've served as a reviewer for lots and lots of manuscripts over the years, which is a typical thing for a scientist, research active scientist to do. And I served as editor for a few journals as well, but reviewing editors, not the editor level of those. In terms of patenting, certainly the dicamba resistance gene technology we patented. That was an education unto itself. Fortunately, As the director of the Center for Biotechnology, I became involved with a national group called the Centers for Biotechnology, which was affiliated with the Biotechnology Innovation Organization, which is the major biotech industry organization. And so I got to serve on the board of directors of not only the Centers for Biotechnology. I served as president of that organization. I also got to sit on the board of directors of the Bio organization and became familiar with a number of the lawyers from various biotechnology companies and heard stories about patenting what you do and what you don't do, which were very useful because when we cloned the dicamba monooxygenase gene and the other two genes associated with that system, I knew how to proceed in working with lawyers to work up a patent, how to write the patent, and to know that it was not going to be an easy thing at that time because the biotechnology patent industry was just arising. So that was a very important experience for me. Again, preparing to defend these patents that we had in a court of law was a very humbling and educational activity that I don't want to repeat. Yeah.
So how long did it take to write the patent application?
So writing the application itself, probably a 3-month process with several iterations of the document. Working with the lawyers to make sure that we covered all areas and putting things in legalese, which is not an easy thing for a non-attorney to do. But it turns out that the university at that time was fortunate to have hired a couple of really good lawyers who were very helpful in that process.
And we're talking about cloning here. And in the public's imagination, back in what years are we talking about? 95 or so?
Yes.
There was a lot of discussion about modifying food. Did any of that come back to you? Did you experience any of that?
Of course. The dicamba monooxygenase gene is used primarily in soybean and cotton. And those have been GMOs from the get-go. So the addition of our gene had little impact on public perception of GMOs. The real fight began with the discovery of recombinant DNA technology that led to the Asilomar conference that set up rules and regulations for how you do recombinant DNA technology. And it was at that point that the fear mongers became very active and were very scared of anything that might change DNA. And most people were not aware of the Asilomar conference, that scientists had gotten together to very carefully and deliberately trace the way forward and to set up rules and regulations on what could be done, what couldn't be done, and if something was to be done, how it should be done to be safe. Those guidelines were terribly conservative. I was ready to do recombinant DNA research about about 9 months before the Asilomar conference was called. So I sat and twiddled my thumbs for actually almost 2 years. It twiddled my thumbs in terms of doing molecular biology, gene cloning, what have you. I did other things during that time that were productive, but it set me back 2 years relative to where I could have been. But that was the price I thought was well worth paying, because what I felt was that the guidelines were very conservative. It should make the public feel safe about what was going on. But I miscalculated the brashness, the non-ethical behavior of people who want to oppose a new technology. I just didn't fathom that people would be so irrational. But, we've learned that lesson all too well.
Well, I think it was the 50th anniversary of Asilomar recently, right?
Yes, yeah.
And the things I've read is that if it happened today, it would have been way more, you could do a whole lot more because it has been proven to be so safe.
Yeah.
And that the original players would have come down on the side of, this is pretty well contained.
Yeah. So even though with, gene editing is new.
Yeah.
And unfortunately, it's being received a little more kindly, but not much more kindly than genetic modification through genetic engineering. What surprises me is that this technology has been around. Genetically engineered crops have been available since 1996. Okay. So that's almost 30 years.
Yeah.
And no ill effects from that technology are documented at all. And yet there's great fear. So the ability to convince people that something should be feared is very powerful. I grew up thinking that logic would prevail, and have learned that.
So did I.
Unfortunately, that's not always the case.
No. Humans are amazing. Let's see. So I guess the next question is, where did you get to funding for your research? And then how did the funding organizations change during your time as a researcher at UNL?
Yeah, so my first grant to do research was at the Fox Chase Cancer Center in 1972. And it was from the National Science Foundation. And fortunately, throughout my career, even while I was in industry, I've had NSF support. So that's been a bedrock for me. Not easy. The competition for those grants was quite fierce, but we were able to maintain our funding levels well. And then when I moved into the genetic engineering of crops area, that was a very tricky one, because there were very few funding agencies that would fund that research. Even USDA had very few funds to fund anything with genetic engineering of crops. Fortunately, there was an organization that was set up first at Purdue and then expanded to all agriculturally related universities, called the Consortium for Plant Biotechnology Research, that worked with USDA to get funds from USDA that could be matched with industrial funds to grant a particular proposal that had long-term potential for payoff. And I was fortunate enough to receive one of those first grants, and then that organization funded most of the early part of my work with the dicamba monooxygenase, the dicamba resistance gene technology. And then after we patented the dicamba monooxygenase, well, even before that, we had some industrial sponsors. And ultimately, after we patented it, that technology, and it was licensed by Monsanto, they provided grant funding for us for a few years to carry on and improve and expand that technology. Yeah.
So the USDA program, is that still in place?
A version of it called FAR that is slightly different, but it's still there and is still an important source of funding for people doing research that has a fundamental aspect as well as an applied aspect to it.
Mm-hmm. And then you have an agreement of some kind that's worked out between UNL and Monsanto. How does that sort of stuff work? How do you get those set up?
Yeah, so it actually it was, I think it's fair to say that we our dicamba monooxygenase technology is one of the very few patented technologies that has found its way into crops from a university. There are certainly lots of genes, lots of plant breeding that has gone into crops that initiated at the university. So most of the biotechnology that has gone into crop improvement has been done by the companies themselves. So we were fortunate enough with a relatively small group to bring this technology into a position where we could show that it worked in plants, in the field, in the crop plants per se. And that made a huge difference in being able to license that technology to Monsanto. And then we worked very closely with Monsanto with the research that we did. We had regular meetings with them to show them what we were doing. And they benefited greatly from that technology, some of which was never published by us. So it was a good balance between the university and the industry in that case.
No, it feels like the ideal that's been pitched at universities, get your products, your research into industry and capitalize on everything, but it's super hard, it sounds.
Yeah. So when you're trying to work at the very cutting edge of what's going on, it can be challenging because you're exploring things, trying to do things that no one's done before. But I found that terrifically exciting as well, to come up with innovative ways to approach a particular problem, to see if it works or doesn't work. And I guess what most people don't understand, I hate to put a percentage on this, but certainly 90% of experiments fail. Okay, because you're working on a hypothesis and you're trying to prove or disprove that hypothesis. And the methods that you're using often are brand new. You don't know if they are working or if your idea is crappy, and that's the reason it's not working. But that getting results, sitting down, analyzing them, especially with a group of students, and saying, why didn't this work the way we thought it would work? What can we do differently? allows the pot to boil, to the juices to flow, and that's exciting and that's fun.
Yeah, absolutely. Yeah, so we've already covered a little bit about the techniques and instruments. How about this one? Was your research ever scooped, or did you and another group tie in publishing the same results at the same time?
Yeah, so we certainly have. I talked about the cloning of the gene responsible for the CIA5 mutation. Well, it turns out that there was a group in Japan that we knew had interest in the gene, but we didn't know they were going after the cloning of that gene. And turns out that they submitted their manuscript and we submitted our manuscript to the proceedings of the National Academy of Science almost simultaneously. And those came out in the same issue.
Oh, wow.
So I wasn't scooped, but
But a tie.
yeah. So I did have, though, during my career, lots of competition throughout my career, and that, if that's part of science, is that there are other people who like to study the same kinds of things that you do, and there's a friendly, sometimes not so friendly competition. I must say that I had one competitor who was especially nasty, and that bothered me for a lot of years until just about 3 years ago. I went to a conference, an international conference in France, where a group of us working in about the same area got together. And I mentioned that my competition with this particular individual had been tough. And everybody else around the table who interacted with this individual reported the same thing. And it turns out that I wasn't treated any more poorly than anybody else. But in fact, most of the people in the field long to be collaborators instead of competitors. And that's led to a big change in the atmosphere of meetings. One of the first meetings I went to, I was trained as a plant biologist, a plant physiologist. And one of the first meetings I went to was a meeting of the American Society for Plant Physiology, which is now the American Society of Plant Biology. But this was held at the University of Massachusetts. And one of the symposium that I went to featured a talk by a Professor Hans Kinde from the Michigan State University, who was very well known, very well respected individual, who gave a talk on a plant hormone called cytokinin. And he gave his talk. There were questions and so forth. And then Folke Skoog, Professor Folke Skoog from the University of Wisconsin, who was the premier scientist in this area, stood up and said, I want to apologize. I want to apologize on the behalf of the U.S. taxpayers who paid for this crap. And so that I take it from talking with some of the more senior people in the field that that was kind of the end of the era of people being nasty at meetings. It was certainly true in chemistry and in physics for years that you were taken to task for lots of things. So meetings now are much more pleasant to go to. Whether they're more effective, I don't know, but they are much more friendly. And I think people underplay the importance of scientific meetings, gatherings. you go not only to learn new things, but more importantly to learn to know the people in the field. And in this day and age, when there are so many different expertise areas of expertise that have to come into any one study, having collaborators, good collaborators, is absolutely essential. And you can't do that well by correspondence or even Zoom calls. Meeting with someone face to face, having coffee with them or a beer makes a huge difference in being able to know them and to know whether you can trust them, to know if they have compatible ideas, approaches. It makes a huge difference in the success or failure of those collaborations.
Yep. No, I agree. Collaborations have increased a lot since the 90s. And I'm not going to say it's the norm, but there are a lot of them, bigger groups attacking more complicated problems. Let's see. Did you publish anything that was ever contradicted, and then how was it resolved?
No, fortunately. We always tried to be very careful about what we published. Everything that we did, every experiment that went into a publication was performed 3-4 times. And if the result, we only accepted the results if they were quite reproducible. Interestingly, when I started out my career in biochemistry, statistics were hardly used at all. Either an experiment worked or it didn't work. And there's a little bit of justification in that because you proceeded much more rapidly, sometimes erroneously, but then late in my career, everything had to be verified through statistics. And that's good. So as long as you're working with good statisticians, and I was very fortunate. We did some genomics work with Steve Ladunga of the genetics of the statistics department that was first rate and wouldn't have been done without him and his statistical analysis capabilities.
Well, these large data sets is where everything's moved, right? So you totally need statistics to know if you found something. A related question is, did you ever publish a correction? And then why?
Nope, no
corrections.
Publish any correction. And again, that goes back to being very comfortable that what we were putting into a manuscript was reproducible. And could be if possible, we attacked the same question from several different directions to get more than just one central line of support for a finding. Yeah.
Okay. Let us then. So the next categories are other career activities, your youth, and then your undergraduate and graduate training. If unless there's anything else you want to say about your award-winning research.
I don't think so.
I think we got a really good idea here. On other career activities, of course, being a professor has a lot of different components to it, and you talked about going to conferences already. So which ones were really valuable to you? So we talked about the biotech directors meeting, but then also scientific conferences.
I think I can honestly say that I never went to a meeting that I didn't think was productive. There were some that had large parts that were boring, but there were always some gems, both in terms of the information that I learned and in setting up new friendships, new collaborators. so I, as my career evolved, the meetings that I went to, my first meetings were the Plant Physiology Society's meetings, and then it was the cell biology meetings, and then the Chlamydomonas reinhardti research community became big enough that we set up our own international meetings. And I helped set up the first of those and ran one at the University of California in Santa Cruz that was the best Chlamydomonas meeting I've ever been to, even though I raised. That's not being modest, but it was a beautiful atmosphere. I don't know if you've ever been to.
I've only seen pictures.
Santa Cruz campus, but it's gorgeous. And it lends itself to informal interactions as well as the formal presentations. And so that set of meetings, and one of the things that I did in subsequent meetings of that organization was to provide a summary of the findings that were presented at each of those conferences. And I found that terrifically fascinating. I had to listen to everything that was said during the meeting, and then put that into a summary. And that was a great learning experience for me. And it made me aware that you need to be careful not to shortcut people and their contributions. Mm-hmm. Yeah. So, but one of the most memorable meetings I went to was at MIT, the Massachusetts Institute of Technology, in the very early days of molecular biology, of gene cloning. This must have been, it was in the early 70s, very early 70s. And this conference was a meeting where Paul Berg and others who were involved in the early days of gene cloning were on hand. And I heard some of the base technologies that then exploded into all of the techniques that were available in molecular biology. And I got to meet and talk with those pioneers. And that was a groundbreaking experience for me. It also let me know that there was some pretty heady competition out there. These were really bright people, and if you wanted to play in that arena, you were going to have to work pretty hard. So an eye-opener in several ways.
Mm-hmm. How about the biotech directors meetings? Did they give you any tools or contacts to help run the facility at UNL?
Actually, to begin with is what are the best methods to run a Center for Biotechnology? What things should you be doing? What things can you do better? Those were terrifically helpful. By the time I became president of that organization, I think in 1994, we were setting up symposia for the bio organization. So we would put together essentially an academic symposium on a particular topic. So for instance, one of the topics when I was president that we presented was on xenotransplantation, using animal organs for human transplants. And that opened a lot of eyes for people in the biotechnology industry. And as you probably are aware, there's been great progress, slow progress, but it needed to be slow, in producing organs from animals that can be used in humans. So I think it's not too many more years that a lack of organ donors will not be such the serious problem as it is now. Mm-hmm. You know, as a quick aside, my grandson, Griffin, when he was born, had an inborn error in his DNA that led to a serious muscle disease that was centered around enzymes in the liver. He received 2 liver transplants. He would have died if he had been born a year and a half earlier, because at that time, a year and a half before he was born, at Duke University, they discovered the gene that caused this disease and were able to diagnose it. He would have gone undiagnosed and not received the liver transplants without that technology. So, I feel I'm a prime beneficiary of biotechnology.
Sure. I think we all are, yeah. Let's see. Sabbaticals. What did you do? Where did you go? And how did that influence your research direction?
Yeah, so I did 2 major sabbaticals. The first was to the University of Geneva in Switzerland with my friend Jean-David Rocher. And that turned out to be a pivotal point in my career. Not so much for the science that I did there, although I did develop some new techniques there. The prime thing was it placed me in Europe, and I did a lot of traveling to other universities and got to know people person to person that I'd only read their research, they had read mine. And that turned out to be terrifically useful. Those contacts really paid off big time for me down the road in establishing collaborations, helping to get things done in the science community. So that was very useful. Then my second major sabbatical was at Stanford University at the Carnegie Plant Biology Research Facility on the Stanford campus. And there, it was a great time to get to know some really top scientists on the Stanford campus and to interact with them. And it was at a time that I needed a bit of a break. I had been going full tilt for about 15 years and without any break at all. And we were able to, for instance, I took a trip to with my sons and son-in-law to Mexico for 5 days. That I'd never had a vacation per se until that time. So that was a great break and a great learning experience as well.
Did you look at any cornfields in Mexico?
Did I look?
Any cornfields?
Actually, yes. Virginia Walbot at Stanford was a maize molecular biologist, and we were good friends. So I did get to see her corn. I wouldn't call them fields; I would call them plots.
Sure.
I did get to see in the Central Valley a lot of almonds and the other crops grown in the Central Valley, which was an eye-opener for someone from the Midwest.
Service contributions. So as biotech director, I don't know how much service you ended up doing, but how did that all work out?
Yeah, so
with the department, with the university, with the college, all that stuff.
Yeah, so certainly as director of the Center for Biotechnology, what was wonderful about that position was that it covered all of the biologists on the campus. That's both East Campus and City Campus. So it was only a few years later that I recognized how important it was for me personally to get to know people on both campuses to help bridge some of the gaps that existed. between the two campuses. So there was a lot of services there. Then as department head, I had lots of different committee assignments on campus as well. Nationally, I served on the board of directors of the Biotechnology Innovation Organization. I served as president of the Centers for Biotechnology. I organized a number of scientific conferences throughout the world. And then within the department, the normal committees of promotion, tenure, the curriculum committee, those sorts of things.
Great. And then teaching. What courses did you teach more often than others? And what are your thoughts about those courses?
Yeah, so I ended up teaching the second semester of advanced biochemistry, junior and senior biochemistry. And the second semester was primarily molecular biology type things. And I greatly enjoyed teaching that course. The only negative that I saw was that it was a large course. We had a lot of students that filled the auditorium and the Beadle Center. And so I didn't get to know many of the students well. And that's the course that I enjoyed most teaching was actually an advanced biotechnology course for graduate students and top-ranked seniors. Only a few undergraduates took the course, but it was limited to 16 people. And it was to introduce graduate students in laboratories that weren't doing molecular biology to the techniques of biotechnology. Some of those students were top-notch students who had a little bit of experience, and it was a joy to work with them. Some of the students knew very little molecular biology, so there had to be a lot of bringing up to speed in terms of the knowledge base as well as learning the techniques. And what turned out was a very nice relationship within that group, and that some of the students who were more capable, who had more experience, ended up helping to teach some of those with lesser skills. And so the learning curve for those students, I have never seen such a steep learning curve for those students that came in at a very base level. So you really got to know those students personally, knew their strengths, their weaknesses, and I'm still in touch with some of those students even today.
Wow, that sounds like a great course.
Yeah.
Let's see. So, anything else about your career at UNL, or? But then we're going to go to your youth and your training after that.
Well, I certainly am very thankful for the opportunities that I had at the University of Nebraska. The university served me well, and I hope I did reasonable service for them.
No, it all sounds really good. So your youth, tell us about where you grew up and what your childhood was like.
Yeah, so I grew up in rural Indiana on a small 3.5 acre farm, diversified farm, just 10 miles south of Terre Haute, near the town of Pimento. So this farm I grew up on had about 1.5 acres of garden. So we had a big garden. We had laying hens and broilers that we raised for eggs and meat. We had milk cows. We had beef steers. We even had some ducks and some goats. So I grew up surrounded by biology and was fascinated with the way plants and animals grew and developed, what they needed to thrive when they didn't thrive so well. And it certainly provided ample opportunity to develop a work ethic. There was everybody in the family was expected to chip in. I also had an opportunity to earn extra money during the summers and helping farmers around doing various chores.
How about 4-H?
Yeah, so most importantly, the school I went to was very small. There were 10 in my graduating class. So that was unfortunate in that I wasn't exposed to a lot of academic areas. And a few of the teachers we had were not very high caliber, but a few of the teachers that we have were terrific. Not so much for what they taught, but teaching us how to live, how to become part of the community, accepting responsibilities, those sorts of things. It was a terrific atmosphere to grow up in, a very supportive community. And a big part of that was the 4-H organization for youth. It allowed us to learn to do certain skills that was fairly minor in retrospect. What it really taught us to do was to learn to take responsibilities and to grow into leadership roles that just wouldn't have existed otherwise. And for me personally, I was able to do quite well in the 4-H organization at the local, state, and national levels. So I earned scholarship monies. I couldn't have gone to college at Purdue without that scholarship money. And then I was fortunate to be selected to give the 1963 report to the nation, the 4-H program, along with 5 other 4-Hers from throughout the country. We presented that report to President Kennedy, members of his cabinet, our congressional representatives, as well as various department heads in Washington. Then we went to New York and Los Angeles to work with the business community. That was a terrific learning experience for me. And we keep intact with all of those members. We just had an anniversary meeting this past year.
So what did you specifically focus on? Did you do animals or did you do plants?
Primarily plants. I had we did poultry, but that was a bit of an aside. Beef steer competition was a big part, but the garden was actually the prime focus. And my first national award in 4-H was for the work that we did in gardening. Yeah.
Awesome.
And that may explain why I became a plant biologist.
Yeah, it seems like a direct connection. But yeah, science and math specifically, it sounds like you didn't get so much of that in school, but you somehow knew you needed it.
Yeah, so it turns out that one of the really good instructors that we had, but we had him only 3 years, was George Walton. I must say that the most important teacher I had as in high school was John D. Turner, my vocational agricultural teacher. He really focused on developing people, okay, and was terrific at it, and played a big role in our 4-H program as well. But on the non-ag side, we had a math and science teacher by the name of George Walton, who had been a former engineer who had retired and did the teaching something to do in retirement. The most memorable course I had with him was geometry. It was my first introduction to what could be called logic. And I love that course. And Fortunately, there were 5 or 6 other students in our school who were top-notch students who were all in that class. And we formed a friendly competition as to who could prove a theorem most rapidly, who could come up with alternative proofs. And it was such an intellectual high that was really formative for me. Yeah.
Yep, it sounds really fun. So let's go to your undergraduate training. And we said you went to Purdue, but like what department, what degrees, what did you experience at Purdue?
It's interesting because I I was told in high school that chemical engineers can make a lot of money. And I kind of like chemistry. So I signed up for chem engineering. Um, but as I said, I was in 4-H, and one of the scholarships that I got was I worked at the state fair in one of the buildings in the horticulture building, helping with displays and helping exhibitors put up their displays. Um, and so my senior year in high school, it turned out that I became eligible to compete for a state fair 4-H scholarship, which actually was quite handsome in terms of the monetary return. And I competed for that and won that. And so it turns out that that was only good in the School of Agriculture. It wasn't good in chemistry. So I quickly switched over to the ag school, and they had a new program called Agricultural Sciences, which on the face of it, you would think would teach you how to be a farmer. But the emphasis was on science. And the courses I took were actually, most of the courses I took were with the engineering students. So I took the engineering math, I took the engineering chemistry, the engineering physics. So a pretty rigorous course of study at Purdue. And that served me very well. But the most influential instructor I had was actually freshman botany. A fellow by the name of Postlewaite, who, as it turns out, was a world famous teacher of botany. This guy was fantastic. And he really inspired me to think about becoming a scientist. Not directly, but indirectly with the way he taught, what he emphasized, and how he led us to challenge the status of knowledge. It was quite an experience. Yeah.
Sounds really great. Did you do undergraduate research?
Yes, I did. As a junior and senior, I worked in the laboratory of Dr. Joe Key, who was a fantastic scientist. And it's actually, he had done his graduate work at the University of Illinois, and it was on his recommendation that I went over to Illinois to work with Dr. John Hansen as my primary advisor.
Yep, that was going to be my next question. How did you choose Illinois?
So it turns out that John Hansen was a plant physiologist who was in the agronomy department, but he insisted that all of his students take their primary coursework in biochemistry, with a smattering of biology thrown in. And so that's what skewed my path toward plant biology and plant biochemistry and plant molecular biology. Well, that was before the days of molecular biology, but so that's the way I got to Illinois and working with Hansen in particular.
Mm-hmm. And so, what was it, 4 years at Illinois, and then start looking for a postdoc, thinking about postdoc. Seems like a left field choice to go to a cancer center, but how did that come about?
Yeah, so actually, I had initially set up to do a postdoctoral work in Wales,
Oh,
in Aberystwyth with Wales, with P.F. Waring, Dr. P.F. Waring, who was an outstanding plant biochemist. Um, uh, but for that, I had a fellowship from the federal government, and times got tough, and those were canceled. So I looked around, and a person that really caught my eye was Abraham Marcus, who had just published his first paper on the wheat embryo in vitro protein synthesis system.
hmm.
Um, and I was doing work on polyribosome formation in corn seedlings and thought, this guy is really doing first-rate research. And he was at that time with USDA Beltsville, the major research facility for the USDA in the United States. And so I approached him, went out to visit. We decided that he decided to hire me. So about 3 weeks before we were set to move to Beltsville, I got a call from him, and he said, I want you to know that I'm no longer at Beltsville. I've taken a job at the Institute for Cancer Research at the Fox Chase Cancer Center in Philadelphia. I can offer you a position there, but if you want to turn that down, there's no hard feelings. And I said, well, I think I want to do it, but let me talk with my wife. And so we decided, what the hey? And it turns out that was perhaps one of the best things that ever happened. Because as I mentioned earlier, the Fox Chase Cancer Center, the Institute for Cancer Research, was an #1 research laboratory or institution with top research going on, fundamental research in cell biology and biochemistry, with lots of opportunity for me to grow. My background was plant biology. So my learning curve was steep, to say the least, but very rewarding. And I got to learn from the best. Yeah.
So probably should have asked you this one a little earlier, but what did you do to merit authorship on your very first paper?
Well, I did the work.
Yeah. And you wrote the paper, the whole thing?
I wrote most of the paper. Yes. So this was with Abraham Marcus, and it was a paper that was based on my ideas. So Marcus was a tough mentor. He was demanding that you do experiments, you set them up correctly, you interpret them correctly, and that served me very well. There were others in his laboratory that found it difficult to rise to the challenge that Abe presented, but I thrived under that, and then he was in his own way very supportive, in a demanding sort of way.
Sure. Tough
So
love.
so I wrote the paper. He did some modifications of the paper, but pretty well as I wrote it.
So yeah, but it doesn't sound like you had much training in writing, right? But you probably have written many reports and
Yeah. So it turns out that one of the negatives of going to a very small high school was I maybe wrote 4 or 5 papers during my whole high school career. And so when I went to Purdue, my first, the only C I had, the only grade other than an A I had in my freshman year was a C in chemistry in English, and I deserved that C because I didn't have writing skills worth anything. So, but fortunately, I had opportunity to write reports and so forth, and so my writing skills improved. They weren't great. And then as a graduate student in writing my thesis, that was terrifically demanding. And so by the time I became a postdoc, I had what I would call fair writing skills.
In retrospect, how far did your career research at UNL research diverge from your graduate and postdoctoral work?
I would say that I evolved dramatically. I wouldn't say I ever switched. It was all an evolution. And I have always been a very strong believer in the power of techniques. That you can't do good science without good techniques. And I love to develop new techniques. Um, I found that rewarding, both just from a fun thing to do. I found it rewarding also because it put me in a position to help others do their research and move the things forward. So I went from being a plant biologist to being a algal molecular biologist to becoming a genetic engineer to becoming a gene editing person. Yeah, thank you for doing this. I think this will be a great service to the university.
I think it's fascinating what a life you've had.
Yeah. Well, yeah, you should get to know some interesting people.
Yeah, yeah. All right. So this is the personal part. And tell me about your family, right? When did you get married? How many kids did you have, if you had any? And then how did that all work out throughout your career?
So marriage was a terrifically important part of my life. I met my wife through 4-H.
Oh.
No big surprise. We were in the same county. I was in the south part of the county. She was in the north part. And through our 4-H leadership activities, we got to know each other. And then I was at Purdue. She decided to go to Purdue. So we dated while I was still there as a senior. And we were married June 6th, 1964, 20th anniversary of D-Day. So I never forget my anniversary. I think she did that deliberately, so I wouldn't. So, but I've been terrifically fortunate. Rita has been terrifically supportive of me. We have 4 children. Our daughter, Emily, came along in 1966. And then 3 sons followed that, John, Derek, and Drew. They have all done very well for themselves, so feel very pleased about that. We have lived, grew up in Indiana, went to Champaign-Urbana for graduate school, Philadelphia, then to the Bay Area, lived in Cupertino in the Silicon Valley, which was an experience for us and certainly our children growing through high school. And then ultimately here in Lincoln. So, yeah, I'm terrifically pleased with the way things have turned out for me and my family.
So the kids, what do they think about all the moves?
Well, the move from Illinois to Pennsylvania was when they were very, very young. So they really only knew Pennsylvania as their home, their first home. the move from Philadelphia to the San Francisco Bay Area was a bit traumatic, especially for our two oldest, who were juniors going into their senior year in high school. That timing could have been better. But as it turns out, they adapted pretty quickly. And our two younger sons, I would say, adapted instantly and loved the environment in the Bay Area. They really flourished in the high competition of the Bay Area. And we were in a community primarily of engineers, being in the Silicon Valley, engineers and entrepreneurs. And so it was certainly a far different environment than we had in Pennsylvania. And primarily positive, but I found the materialism in the Bay Area a bit off-putting. The me first attitude of most people was not the way I grew up. So took a little bit of doing. But there were some valuable lessons for me in making that move. I grew up in a community where it was looked down upon to fail. And so your goal was to be successful in in the endeavors you undertook. For the first time in my life, I was in a business environment in the Silicon Valley where it was okay to fail as long as you had gained experience and could learn from that experience. That was completely foreign to me. And it, I think, gave me the courage to be more aggressive, more risk-taking than I would have been in my science. I made some pretty important decisions in changing the way I was approaching the science that I was doing that I might not have done without that experience.
And so, yeah, before retirement, what did you do for relaxation and entertainment? Just found out you only went on one vacation, but tell us more about what do you do outside of research?
Yeah, so I stopped my scientific collaborations about 2 years ago. So I continued those up until that time. So my retirement was a bit gradual, but I became involved with a number of activities. We live at the Grand Lodge retirement community, and I've been involved with a number of projects here. Became heavily involved with the Emeriti and Retirees Association of the university, and that's been rewarding in many, many ways. Then my avocation is genealogy. I have been fortunate, actually. I knew relatively little about my family histories, but for all four grandparents now, I've done genealogy and have been able to go back several centuries in all cases. Some in the Weeks family and the Kester family, all the way back to the Norman Conquest and before. So that consumes a lot of time. There's a lot of information out there now. Unfortunately, a good portion of that is not correct. So doing documented findings is a challenge, but it can happen. So.
All right. Well, thank you very much for talking to me. Anything else?
No, thank you again for doing this. I think in the future, people will appreciate getting to know people who were part of the University of Nebraska and what made it tick and what made it successful.
Yeah. Thank you very much.
Thank
All
you.
right. Great talking to you.
Yes, thanks for the opportunity.